metastasis. They can contribute to the immunosuppressive microenvironment that excludes T cells in the first place. They can also secrete different cytokines or growth factors that recruit blood vessels to feed the growing tumor. So just from a really big picture perspective, wouldn't it be great if we could very specifically turn on and off different macrophage behaviors and tune macrophage signaling to be exactly what we want? Wouldn't it be good if we could rewire macrophage signaling? Okay, now in order to do this, we're going to need a lot more information about the basic signaling pathways that macrophages use to determine how they should behave. So you can think of macrophages as sentinels of the innate immune response. They're constantly around surveying tissues, looking for signs of injury or infection. While they're out on patrol, macrophages are integrating many, many different signals to decide how to respond or what their output should be. To add more complexity to this, they're not just measuring the binary presence or absence of different signals, they're also measuring how much of a signal is present. So my approach is to isolate specific inputs or sets of inputs and figure out how the macrophage measures and adds all of these different signals. And then long term, I aim to build up the complexity of this system to predict how a macrophage will respond to a target and tune this response with rationally designed therapies. Okay, so now let me tell you a little bit about the approach that I'm taking. So I think this requires some synthetic biology. When a macrophage encounters a target in vivo, that target can vary in size, shape, stiffness, and many other parameters. Most concerning for me is that that target will expose many, many different signals that can all inform macrophage behavior. We don't even always know the full complement of signals on that target surface. So without quantitative information about what signals the macrophage has received, it's very difficult to understand what they're responding to. Yeah, so somehow the macrophage has to integrate all of these different signals to select its response, and in order to dissect this process, we need quantitative information. So instead of this complicated target, I've decided to start with a reconstituted, controllable engulfment target. So I start with a silica bead. On that bead, I build a supported lipid bilayer, and then on that bilayer, I can conjugate a defined signal or defined set of signals and watch how the macrophage responds. Okay, so today I'm going to tell you about one specific problem that the macrophage faces. How does it decide what to eat? This is actually quite a problem for the macrophage because they are constantly bombarded with positive stimuli. So IgG, for example, which I'm showing here, is present at mg per mil concentration in healthy adult plasma. Somehow, in the face of all of this positive stimuli, macrophages need to distinguish between healthy cells that have transiently or non-specifically bound to some stimuli and true pathogens. It's essential that a macrophage make this decision correctly and robustly ignore healthy cells, or else the organism will develop autoimmune disease. It's equally important that the macrophage rapidly recognize and engulf pathogens and debris to keep the body free from disease. So the question that kind of is guiding my way right now is, how does the macrophage measure and add all of the signals on the target cell surface? Okay, so let me show you what this assay my standard assay looks like. So like I've said, I coat silica beads and supported lipid bilayers and then add a defined complement of signals. So here I've conjugated different concentrations of IgG to the beads. And IgGs are antibodies that signal for the macrophage to engulf that target. What you can see is that the macrophage ignores low densities of IgG, but engulfs these high densities of IgG. I think this brings up a really interesting and important question, which is, how does the macrophage ignore targets with a little IgG or with subthreshold stimuli? So I think hopefully by now it's very obvious to you that if a single antibody activating a single receptor led to engulfment, that would be a total disaster. So instead, engulfment requires a coordinated response from many receptor-ligand interactions. So what does this coordination look like and how is it achieved? Now, to start to answer that question, let me first tell you a little bit about the signaling pathway that macrophages use to respond to IgG. So when IgG is bound to a target, it's recognized by the Fc receptor in the macrophage. Upon IgG binding, the Fc receptor is phosphorylated and triggers an intracellular signaling cascade that eventually leads to actin reorganization and phagocytosis. Now, one of the great things about this synthetic system that I'm using is you can take exactly what's on your three-dimensional target and put it on a two-dimensional glass cover slip. Then I can use turf microscopy to look at just the macrophage target interface with really high temporal and spatial resolution. So this is a really good way to visualize receptor dynamics, or the very first events in the phagocytic signaling cascade. Okay. So here I'm showing you some data from this type of assay. In this movie, the IgG is labeled in black, and you're watching from below as a macrophage comes and lands on a supported lipid bilayer. It's just one macrophage. Okay, so what you can see is as the macrophage lands, the IgG is gathered into these nanoclusters, and these coalesce into larger scale micron clusters. We know from previous literature that these IgG nanoclusters recruit downstream effector proteins. So it's been assumed that these are key regulators of macrophage signaling, and maybe they help in the coordination of different receptor-ligand interactions. However, it's been very difficult to directly perturb these clusters without affecting other parts of the signaling pathway. So this really hasn't been directly demonstrated. So luckily, when I was at UCSF, I had the opportunity to work with a really fantastic graduate student, Nadia Kern. And Nadia was just as interested in this question as I was. So what she wanted to do was develop a system where she could quantitatively perturb these clusters and measure the effect. So we had a series of questions when we started this project that I'll walk you through during the rest of my talk. Sorry, I should have mentioned, I purposefully made this talk a little bit short, so please feel free to interrupt me. I'm happy to take questions at any point. Okay, series of questions. So first of all, does receptor clustering affect engulfment? It's a very basic question. We think yes, but we've got to check first. Then second, what constitutes a cluster? A cluster is a kind of vague term, so can we be a little bit more quantitative about how many molecules we're talking about and what sort of organization? And then finally, for us, the real heart of the matter, why are clustered ligands more potent? How does this enhance signaling? Okay, and there were a couple of things that really positioned us to address this question. So the first thing that was lucky for us is that our colleagues at UCSF, Sean Douglas and Ray Dong, had developed a DNA origami technique to pattern ligands. So I'm showing you their DNA origami structure here. It's a 60 nanometer by 60 nanometer pegboard. We call it a pegboard because there's 72 ligand positions on the top surface of this origami that can all be controlled independently. So they could either have the same signal the same sequence exposed or different sequences. They could have no sequence or have a sequence. Then on the corners of this pegboard, there's a fluorophore that we can track it. And then on the bottom, there's biotin, so it can attach to our supported lipid bilayer. Okay, the other piece of luck that put us in the right place at the right time to answer this question is that I had just finished a paper describing the modularity of the Fc receptor. And what I've been able to show is that replacing the extracellular domain of the Fc receptor by anything that recruits the intracellular domain to the targets, to the site where the macrophage is contacting the target is sufficient. So you don't actually need specific extracellular domains, you just need to target the intracellular domain to the synapse. So what Nadia did is she redesigned this endogenous system, replacing the extracellular ligand receptor interaction with DNA. So basically the way this receptor works is the extracellular domain has been replaced with a SNAP tag, which binds to a benzylguanine functionalized DNA receptor strand. This receptor strand has 13 base pairs complementary to a DNA ligand strand, which is attached to the bilayer. Now, for anyone who's familiar with the FCR family for any reason, you might notice that the stoichiometry here is a little bit different. And native IgG will recruit 2 intracellular signaling chains, whereas this DNA-based system requires 2 ligand strands to recruit 2. So it's a 1-to-1 stoichiometry instead. Okay, so we already thought this was really cool because now we have complete control over ligand density and affinity on the target cell surface. So Nadia did a lot of things to validate this DNA receptor and show that it functions just like the endogenous system, but I think the key thing is that it works equally well in triggering engulfment. So here I'm showing you macrophages engulfing beads either coated in single-strand DNA or in IgG, and the same number of beads per cell are engulfed. Okay, so now that we have a DNA-based engulfment receptor, now we're able to use this DNA origami structure to pattern receptor-ligand interactions. So basically what we do is we add many, many DNA origamis to the supported lipid bilayer surrounding our target. And that means that the DNA origamis provide ligands across the surface, and the nanoscale organization of all of these ligands is being controlled. Okay, so now this allows us to ask our very first question: does receptor clustering affect engulfment? So to answer this, Nadia designed a number of different origami pegboards exposing different numbers of ligands. So in these schematics here, the gray dots mean that there is no ligand being exposed, and then the pink dots mean that there is a ligand. So she's got clusters of 2, 4, 8, 16, and 36. Now, to keep the total number of origami boards and the total number of ligands on her beads constant, she's diluted out each of these clusters with the appropriate amount of empty DNA origami boards, or dummies. So again, what she's done here means that there's the exact same number of DNA origamis and the exact same number of ligands on each bead. It's just the organization of these ligands that's changing, just the way they're distributed into clusters. And so what she found is that clustering does in fact enhance engulfment. And I think you can see that this already starts to address our second question: what constitutes a cluster? So what Nadia found is that engulfment peaked when there were clusters of 8 ligands or more. Adding more than 8 didn't really provide a boost in the engulfment signal. And what she decided to do is focus in on this 4 ligand condition because it was almost just as good as the 8, and we thought it would be very sensitive to perturbations in ligand spacing. So the next thing she did to ask how tightly clustered these ligands need to be is generate different origami boards with different spacings between the ligands. So the first one she made is this 4 tight origami board, where the 4 ligands are in a cluster as close together as we can get them in the center of the origami board. Then at the opposite end of the spectrum, she's generated this 4 spaced board where the ligands are as far apart as we can get them on the origami board. And then she's also made an intermediate 4M condition. And what she found is that while the 4M and 4S boards could trigger some engulfment, the 4 tight board with the tightly clustered ligands was much more potent. So tightly clustered ligands, ideally within 7 nanometers of each other, is ideal. Okay, so all of this is really great, and we're very happy with how rigorously we were able to define this system. But what we really wanted out of it is a way to address this question, which is how or why are clustered ligands more potent? Okay, so to answer this question, Nadia basically set up a ton of time lapses of macrophages encountering beads. And because these origamis have fluorophores attached to them, we could basically label them in different colors, put them on different beads, and then add those beads to the same macrophage. And so that is just what I'm showing you here in this movie. The 4 tight origamis, the tight clusters are in orange, and the spaced are in blue, cyan, teal. And what you can see is that the macrophage is preferentially engulfing these orange beads. Okay, now to quantify this, what we decided to do is break the engulfment process into discrete steps and then quantify each step. So the steps that we defined are bead capture, when the macrophage catches the bead; engulfment initiation, the first time we see signs of the membrane wrapping around the target; and then engulfment completion, when that bead is totally internalized. So when Nadia quantified these different parameters, she didn't really find a difference in bead capture. They were captured about the same. But what she did see was a significant delay in engulfment initiation after that bead was captured. And in fact, often for the 4 spaced origamis, initiation completely failed. However, if the macrophage was able to initiate engulfment, the time to completion was about the same. So once engulfment was triggered, it proceeded as normal. So what this really told us is that there's a very specific defect in the initiation of engulfment. Okay, so of course we weren't quite satisfied with that, and we wanted to figure out why there was a defect in initiation. And so what we decided to do is start with the very first molecular events of the phagocytic signaling cascade and work our way through the pathway. So to start with, we have origamis on a bilayer. The first step is receptor binding. And Nadia didn't see any difference in receptor binding. So in these images I'm showing you, there's a 4 tight bead that's not labeled where this orange asterisk is, and there's a bead with the 4 spaced origamis where the teal asterisk is. So the very next step in the phagocytic signaling cascade is receptor phosphorylation. Now, it's a little bit difficult to monitor receptor phosphorylation in vivo, and so what we decided to use is a biosensor. We basically took the tandem-SH2 domain of the next component in the cascade, which is SYK, just the tandem-SH2 domain of SYK. Well, SYK is a kinase and has a lot of intrinsic regulation. This tandem-SH2 domain is inert, and based on our ex vivo experiments, it's recruited very, very rapidly to phosphorylated receptors. And so what you can see is that there's already a huge difference between the 4 tight and the 4 spaced origamis. So the tandem-SH2 domain is recruited very well to this contact with a tight clustered bead, and it's not recruited to the 4 spaced bead. So this already tells us that there's a difference in receptor phosphorylation based on how ligands are clustered. We found that this difference was propagated throughout the phagocytic signaling cascade, and there was a downstream difference in PIP3 and actin as well. So overall, what we concluded from this is that ligand clustering enhances receptor phosphorylation. And of course, we still want to know how ligand clustering enhances receptor phosphorylation, and Nadia is working on that. Okay, so hopefully I've convinced you that macrophages make very sophisticated measurements about the targets that they encounter. Tightly clustered ligands are able to more potently trigger receptor phosphorylation. What we think basically is that this hyperlocal density measurement allows a macrophage to distinguish between a lowly opsonized target and a highly opsonized target. For a lowly opsonized target, it's very unlikely that there will be 2 antibodies bound right next to each other, but for a highly opsonized target, this is much more likely. I also think that this is really interesting to think about when we're designing therapeutic antibodies. So many cancer immunotherapies are in fact antibodies, and they work often completely or partially through Fc receptor function. Now, we still have a lot to learn about what makes an antibody effective at activating the Fc receptors. So there's some parameters that we know from studying protein-protein biochemistry. For example, we know that antigen affinity matters quite a bit. We know that isotype and glycosylation pattern on the antibody affect which of the Fc receptor families are most potently activated. Then the field is expanding to consider some other more biophysical properties of what makes a good antigen. There was a very nice paper last year showing that antigen height can dramatically affect how effective an antibody is. Then I'd like to add to this list antigen pattern. Nadia's data clearly shows that tightly clustered ligands are more potent activators of engulfment. Okay, and then I just want to wrap up by saying I'm looking for rotation students, so if you're interested, please come talk to me. In my lab, we're going to continue working on phagocytosis. I think there's a lot of really interesting questions about IgG still, as well as many other eat me and don't eat me signals. So we have a collaboration with Denise Montel's lab, looking at how we can enhance phagocytosis of cancer cells. We have a collaboration with Max Wilson's lab, trying to make basically a light-induced phagocytosis system where we can shine light on cells and make them eat. Then in addition, IgG can activate other downstream signaling pathways like inflammation. And a lot of these could be really beneficial in the cancer immune microenvironment. But there's a lot of questions that we don't understand, even more than there are for engulfment. So for example, is engulfment of a single bead sufficient to activate inflammation, or does it require a higher or lower amount of IgG? So I'm planning to use live biosensors for transcriptional networks to see how they respond to different targets. So long term, I think this will help us very precisely designed strategies to activate specific macrophage functions. Okay, and then the final thing that I'd like to do in my lab is expand these same tools and techniques to cover other signaling systems. So there are many other macrophage signaling pathways that have shown promise as an immunotherapy target, and often we know very little about the cell biology of how these pathways work. So how are the signals measured and integrated? And I think it will be very interesting to expand what we're doing in the future to examine some of these pathways. And I have some ideas, but I would love to talk to anyone about what you think would be interesting. Okay, and so with that, I would just like to wrap up with some acknowledgements. Most of the work, well, all of the work that was shown here today was done in the Vail lab, and most of it was done by Nadia Kern, who's just really awesome. And then, of course, now I'd like to also thank my colleagues at UC Santa Barbara, and I'm so happy to be here. So, any questions?
I'll ask a question if that's okay. Hi, Megan, great talk. I was wondering if you could talk about how the one-to-one stoichiometry versus what you would expect normally in the endogenous condition would change your interpretation of your results for the endogenous condition? Or do you think it's just something that do you think you're recapitulating exactly what's going on, or?
Yeah, so we think we're recapitulating roughly what's going on. It is a definite caveat, but basically we initially thought that we would basically just divide our results by 2, and that would be what accurately recapitulates the endogenous system. But in fact, it might be a little bit more complicated because Nadia's experiments that I didn't talk about suggest that the reason the cluster matters is that it forms a zone of exclusion for these phosphatases that normally deactivate the receptors. So it may in fact actually be the size of the physical size of the cluster that matters. So yeah, we'll need to follow up on that with further experiments, but given that we can't pattern antibodies at all, we thought this was a pretty good start.